Yes, and then I welcome everybody to this academic session in which Qiuting Yan will defend her academic thesis called The Dynamics of Thrombin Generation. A special word of welcome, of course, to the candidate, far away, but still here, and to the supervisors, Professor Ten Kate, Dr. De Laat, and Dr. De Laat-Kremers. I will welcome and announce the opponents when they ask the questions. But before we go to the actual opposition, I would ask Qiuting Yan to give a summary of her work. The floor is yours.
Dear Prorector, dear members of Corona, dear family, friends, and colleagues, thank you for your attending the defense of my PhD thesis, which is entitled The Dynamics of Xiaomi Generation. In the next 15 minutes, I will present a brief summary of my thesis. When our blood vessel is damaged, our body will react instantly, try to seal off the damage at the site of injury and then stop bleeding. This is done by forming a clot made of protein called fibrin. When we dive into the process of fibrin clot formation, we see that this relies on the function of many plasma proteins and blood cells. The damaged vessel would expose some substance that is normally located under the vessel wall, followed by adhesion, activation, and aggregation of platelets, and expose some. This substance can also triggered by activation of proteins in the coagulation, and this leads to the generation of the enzyme called thrombin. Thrombin is the central enzyme in the coagulation cascade, and it directly cleave fibrinogen into fibrin, and therefore induce the clot formation. Then the bleeding stop. My thesis involves in these two components of hemostasis, platelets, and coagulation cascade. Platelets play a major role in localizing and controlling the burst of thrombin generation and subsequently clot formation. Activated platelets expose phosphatidylserine on their outer members to support thrombin generation. The generation of thrombin can be triggered by intrinsic and extrinsic coagulation pathway. And when thrombin is generated, it will under strict regulation by anticoagulant pathways. The main anticoagulant pathway involves thrombomodeling dependent activation of protein C into activated protein C and the tissue factor pathway inhibitor. And we also have natural plasma thrombin inhibitors, antithrombin, and alpha-2 microglobin. A proper thrombin generating ability is crucial for normal coagulation. To measure this ability, a thrombin generation assay or TG assay is often used. As many people know, the thrombin generation based on the CAT method, and it can be measured in platelet poor plasma, platelet rich plasma, and whole blood. Plasma whole blood can be triggered by addition of tissue factor with the presence of phospholipid, and then prothrombin converted into thrombin, and subsequently thrombin cleave the specific substrate to generate fluorescence. Thrombin generation curve shows the change of thrombin concentration over time in the plasma and reflects the whole process of thrombin activation and inactivation, and therefore give insights into overall function of the hemostatic system. And the thrombin generation curve can be characterized by several parameters: the lag time, velocity index, peak height, time to peak, and ATP. Thrombin generation assay have many potential clinical applications. it could be used to predict the thrombosis and the bleeding risk, and it can also be used to monitor the anticoagulant therapy and to monitor the treatment with the transfusion products. Xiaomi is a balance. Xiaomi generation is a balance of pro and anticoagulant process. So Xiaomi generation is the net result of pro-Xiaomi conversion and Xiaomi inactivation. You see the normal Xiaomi generation curve, and you can see what happens when this balance is disturbed more towards a bleeding or thrombosis. When pro-Xiaomi conversion is low, and Xiaomi inactivation is higher, the Xiaomi generation decreases, and when pro-Xiaomi conversion is higher and the Xiaomi activation is low, the Xiaomi generation increases. So we developed a method called Xiaomi dynamics to study the balance of pro-Xiaomi conversion and Xiaomi inactivation. First, we measure the Xiaomi inactivation by anti-thrombin and alpha-2 myoglobin, and you can see here, and the formulas that we used to calculate. We're using computational modeling to calculate pro-Xiaomi conversion, and it can be calculated from the Xiaomi generation curve and the Xiaomi inactivation. You can see the pro-Xiaomi conversion curve. This looks similar to the Xiaomi generation curve, but it is really pro-Xiaomi conversion curve. Here you can see we also have several parameters to quantify the Xiaomi dynamics. You can see the PC total, that is the total amount of pro-Xiaomi conversion converted, and we also have the PC max. It is the maximum rate of pro-Xiaomi conversion, and Xiaomi inactivation focuses very much on the part of inhibition of Xiaomi over time, and this inhibition is mainly done by Xiaomi inhibitors, anti-Xiaomi and alpha-2 myoglobin. This can all bind to Xiaomi and therefore make less Xiaomi present in the plasma. Especially, we can calculate Xiaomi and anti-Xiaomi component and Xiaomi and alpha-2 myoglobin component, so we can get much more information on the pro-Xiaomi conversion and Xiaomi degradation by using Xiaomi dynamic analysis. One of the main focus of the thesis is the investigation of Xiaomi dynamic analysis in Motifs. In chapter 2, I used the Xiaomi dynamics method to decipher the coagulation profile. In chapter 4, I investigated the use of Xiaomi dynamics method on the fully automated Xiaomi generation analyzer STG media. And in chapter 5, I validated the Xiaomi dynamics method for Xiaomi generation in platelet-rich plasma. And finally, I studied the dynamics of Xiaomi generation in HIV-positive individuals and the effects of the treatment they received. So this thesis focuses on the application of Xiaomi dynamics in fundamental and clinical studies. In the past, the Xiaomi dynamics have been studied in multiple clinical settings to study the balance between pro and anticoagulant mechanisms. So it is of interest to study the contribution of specific coagulation factors to the parameters. We did a series dose response experiments to study the effect of coagulation factor levels on the Xiaomi dynamics. And finally, we found that pro-Xiaomi conversion, either PC total or PC max, are mainly depend on the pro-Xiaomi and factor levels, and Xiaomi and anti-Xiaomi complexes depends on the prothrombin and the factor levels also, and the thrombin and alpha-2-myoglobin complexes depends on the antithrombin and alpha-2-myoglobin levels, and the thrombin and the thrombin decay constant depends on the antithrombin and the fibrinogen levels. As many people know, the cat-based thrombin generation assay is a semi-automated assay. That means when you do one thrombin generation in one laboratory and when you measure the sample in another laboratory, you may get different results. So this calibrated thrombin generation method that we normally use is not optimized. So Stago introduced the STG media several years ago. The STG media is also a method to measuring thrombin generation, but it is fully automated and standardized. Therefore, this method can be used in clinical routine laboratories with a fixed protocols for specific disease and less sensitive to human error and highly accurate pipetting with device. The aim of this chapter is to measure thrombin dynamic using STG media. We measure the thrombin generation using three commercial reagents for STG media, STG bleed screen, thrombo screen, and drug screen in 112 healthy individuals. Then we use the thrombin generation data on the STG media as the input for thrombin dynamic analysis. And the right graph, we show that women using oral contraceptive have a higher thrombin dynamics parameters compared to the men and the women without oral contraceptives. Thrombin generation can be also measured in platelet-rich plasma to study the role of platelet on the coagulation, and the measuring thrombin generation in platelet-rich plasma can be used to study the platelet disorders and to study the platelet drugs that affect the thrombin generation. In this study, we collected 117 healthy subjects, and the platelet-rich plasma thrombin generation was measured undiluted and dilute to a varying platelet count. We found that when platelet increases, the rate-related parameters increases. However, the amount of thrombin formation and the total amount of prothrombin conversion did not increase anymore when platelet reached 150 billion per liter, indicating that platelets increase the amount of prothrombin that is converted and shorten the time until the thrombin production starts. HIV is an enveloped retrovirus that causes HIV disease and acquired immunodeficiency syndrome. Currently, the combination antiretroviral therapy effectively suppresses the HIV replication as related morbidity and mortality are largely reduced. However, more and more patients are suffering from the comorbidities such as cardiovascular disease. So in this study, we collected 55 healthy controls and 189 HIV-infected individuals, of which 96 were treated with abicavir and 93 were treated with tenofovir disoxyl fumarate or TDF. We found that the patients using abicavir had a higher prothrombin conversion compared to healthy controls and the patients treated with TDF. The increase of prothrombin conversion may be associated with a higher risk of thrombotic events. So in summary, in this thesis, we further explored the biological modulators and the clinical relevance of Xiaomi dynamics. In platelet plasma, we determined the major influence of Xiaomi dynamics parameters in platelet plasma, and we validated the use of Xiaomi dynamics analysis in a more automated way on the ST-Geneva device. And in platelet-rich plasma, we validated the use of Xiaomi dynamic analysis, and we show that the platelet count mainly affects the rate of Xiaomi conversion rather than affecting the total amount of Xiaomi formed. We also established the reference values for Xiaomi dynamics in platelet-rich plasma. In clinic, we show that HIV-infected individuals treated with a drug that was linked to myocardial infarction have a higher Xiaomi conversion than controls and other HIV-positive subjects. Thank you for your attention. I give the work back to the pre-rector.
Yes, thank you very much for this summary. And I will go to the actual opposition. And the first question will be asked by Professor Heemskerk. He's an emeritus professor of cell biochemistry of thrombosis and homeostasis at our own university. Professor Heemskerk, the floor is yours.
Thank you, Robert. Dear candidate, let me first start to congratulate you with your very nice thesis on thrombin generation and thrombodynamics. I very much like reading it, and particularly to see how well the field has been developed. And of course, in my congratulations also include your promotion team, who has been instrumental in developing of the field and in getting your thesis done. And to understand how you actually have carried out your work, I deduce that from your propositions 9 and 10. If I understand it well, is you are not comparing yourself with a fast horse, but you are rather a person who learns and thinks and differentiates before doing the actual experiment. And I think this is highly admirable. However, my job is not to praise you, but to ask you some questions on aspect of your thesis. So my first kind of general question is, in the two essays you have been using, thrombodynamics and thrombin generation, what do you think are the most important parameters? Are these the early parameters or the late parameters? So is it the start of the curve, or is the total curve that you think are most important?
Dear highly esteemed opponent, thank you for your kind words and thank you for your question. Regarding your question, you asked me what is the most important parameters in Xiaomi dynamics. I think the most important parameters in Xiaomi dynamics is the total amount of Xiaomi converted. These parameters is very similar to the ATP in Xiaomi generation, and these parameters quantify the total amount of Xiaomi conversion. And from this parameter we can know how much Xiaomi converted to the Xiaomi. So I think this parameter is very important. Is the most important in Xiaomi dynamics.
Okay. That's a good point. So you prefer the PC total over the PC max. Actually, that's what you're saying. From that, I would like to go to page 39. My question on chapter 2, page 39. And then if I look there at the panels D and G, if it is the variation of factor 5 levels and factor 10 levels, I see that actually that the PC max is more sensitive to the tissue factor concentration than the PC total. So in some way, that just to me, if you let's say that the PC max is a kind of more sensitive parameter to look at effects of triggering. So my question is actually, what is your explanation for the fact that the PC max is more sensitive to the tissue factor concentration than the PC total?
Actually, the PC max is sensitive to the factor 5 and factor 10 levels. So we can say the left columns with increasing factor 5 levels, it doesn't make a big differences in the PC total, but when your factor 5 level is from 10% to 100%, this finding is in line with the reports. The very low factor 5 level are sufficient to support hemostasis. And when your factor level is very low, even lower than 10%, you have a low Protrom conversion, either PC total and PC max. Actually, in clinical settings, patients suffering from factor 5 deficiency usually present a bleeding phenotype and prolonged APTT or PT. And for factor 10 levels, factor 10 is the most important members of Protrom complex. You can see in the right panel the effector is largest difference for a very low levels, but there is the curve are bit less steep with the factor 10 level increases. And also in practice, the patients with factor 10 the deficiency of factor 10 have a severe bleeding phenotypes similar to the hemophilia patients with a deficiency of factor.
I agree. There's a very nice, very clear description of the experiment of the curves you show. But let me come back to this figure later. But let me first switch to page 121. There you look at platelets. So that is table 3 and page 121.
Yes.
There I see that the early parameters, lag time, peak, time velocity index, and the prothrombin PC total are very sensitive to the presence of platelets. So that suggests to me that in the early especially in the early parameters, there is a high effect of the presence of platelets. So what is your understanding? Why do you get a higher thrombin generation if there's more platelets present? And a higher thrombodynaamics if there's more platelets present? So what is the role of platelets to enhance the prothrombin conversion and to enhance the thrombin generation?
If you understand correctly, your question is with the increase of platelet. So what we can say from the Xiaomi generation and the Xiaomi dynamics?
Yeah.
We did experiment in these studies. In this study, we found that increasing platelet count, the total amount of ETP and ETP, the rate of Xiaomi generation, for example, velocity index and peak height increases linearly with platelet increasing of platelet count. However, the ETP reached the maximum level at the platelet when the platelet reach 150 billion per liter.
Yeah.
And also in thrombin dynamics, we also found the similar results. When platelet count reach 150 billion per liter, the PC max continue to increases, but the total amount of thrombin converted reached the maximum level.
This is a very clear explanation. Then I have two very short questions to finish.
Very, very short, Professor Heemskerk, because
so the first one is at this page, which tissue factor concentration have you been using for this table to look at platelets? Was that 1 picomolar or 5 picomolar tissue factor?
5 picomolar tissue factor. We use
I think you use 1 picomolar. It's stated somewhere. But then my final question is: would you agree that the presence of platelets compensates for a high tissue factor concentration? So that if you add platelets, that you do not need to add a very high concentration of tissue factor anymore. Have you thought about it?
Sorry.
Because if you add platelets, you get a higher thrombodynaamics. If you add more tissue factor, you get a higher thrombodynaamics. So platelets, in fact, compensate for the presence of high tissue factor.
Platelet can compensate a higher tissue factor.
Is it different question?
I think we should. I think the answer is not yet very clear to the candidate, so we should leave it at this.
Yeah, thank you.
Thank you, Professor Heemskerk, and I hope you will have an opportunity to discuss this further afterwards. Because I would now like to give the floor to a guest from another university, and this is Professor van Gorp. We are always happy that there's opponents from elsewhere because it attests to the importance of the work. Professor van Gorp is a professor of medicine at the Department of Viral Science at Erasmus Medical Center in Rotterdam. Please, your questions.
Thank you, Mr. Rector. Well, first of all, Ms. Candidate, I'd like to congratulate you with the thesis that's in front of me, and of course also your promotion team promoter and co-promoters. But saying that, I'd like to discuss some points with you, and I think the test you investigated, the thrombin generation test, may have a use in clinical practice. It has. But for me, still, it's a question how to implement the test. I do have a background in infectious disease. What role do you see for this test if you are treating patients with infectious diseases in general? You may specify it if you like. Can you give me an advice on that? How would you implement this test in clinical practice in the perspective of infectious disease?
Dear highly esteemed opponent, thank you for your kind words and thank you for your question. If I understand correctly, do you mean how do we use thrombin generation in the clinical settings? Yeah, actually, we already have many clinical studies on thrombin generation. A good example is to use thrombin generation on liver cirrhosis, because liver cirrhosis is a very difficult disease, and patients with reduced of pro and anticoagulant factors, and people may are suffering from the bleeding or thrombosis. In conventional clotting assays, such as APTD or PTR prolonged, indicating that a patient have a higher risk of bleeding. However, from the thrombin generation, the result of thrombin generation, we found that the thrombin generation rebalanced in the patient liver with liver cirrhosis. Yeah. That is good part.
Okay.
Yeah.
Well, if you look to infectious disease, and you already mentioned, then infectious diseases may be complicated, serious viral sepsis and bacterial sepsis by either thrombosis or bleeding, or maybe a combination. And there may be different reasons for bleeding. It may be a consumption coagulopathy. It might be that there is a decreased production of coagulation factors if there's liver insufficiency. And also for thrombosis, there are different mechanisms. Can we use these thrombin generation test in all situations, or do you have to keep in mind what is the cause of, might be the cause of bleeding or thrombosis? So is it one size fits all? We can use this test in all conditions, or do we have to keep in mind what is the cause of bleeding or thrombosis?
I think we can use the Thrombin generation in bleeding and thrombosis. For bleeding, I think the lag time is a good parameter because normally we use APTT or PT to detect the bleeding. And I think the lag time is also a sensitive parameter to detect the bleeding. And for thrombosis, there are already many clinical studies to measure the Thrombin generation on thrombotic events. For example, we can use Thrombin generation. Thrombin generation on the in cancer patients, we can use Thrombin generation to estimate the thrombotic events. Okay. And and we can also use,
Okay, due to time, maybe one more question then, because that's in line with what we are discussing. Does the test has to be validated for every condition, or is that not necessary? What is your idea about that? So need the test to be validated for different conditions.
So could you repeat your question again?
Yeah, well, we were talking about different causes of bleeding and thrombosis, but do we need to validate the thrombin generation test for every single condition to be sure that the results are well, for practical use? So is validation necessary for all these conditions? Well, that's yes, maybe. You understand what I mean, or?
If I understand correctly,
I think you replied already about on it, also mentioning that the thrombin generation test has been validated in cancer patients, as you mentioned. So that's what I mean with the question. Do we need to validate the test under for all these different patient groups?
Yeah, actually, we need to validate in clinical studies for the Thrombin generation in these clinical settings. Yeah.
But there's some time left. Another interesting chapter in your thesis.
I'm sorry to interrupt, but I think time actually is.
Oh, that's a pity. That's a pity. Okay, I give the word back. Thank you very much.
No, thank you. And for the next question, I again welcome an external opponent. We are honored to give the floor to Professor Waal, and he's a professor of vascular medicine in the Division of Region and the Regional Competence Center for Rare Vascular and Systemic Autoimmune Diseases at the Centre Hospitalier Universitaire in Nancy, that is in France. The floor is yours.
Thank you, dear candidate. I hope you can hear me quite clearly. Congratulations for your work. My first question will be about platelet-rich thrombin generation and PC conversion and proton conversion, and I was interested in the work you did on the reference values, which may be very useful for future studies. My first question is, what from your point of view, are the advantages of platelet-rich plasma to study thrombin generation and thrombin dynamics?
Yeah. Yeah. One of the lack of opponent in platelet plasma is a component of platelet plasma Xiaomi generation is lack of platelet, and platelet is important in on the coagulation. So measuring platelet-rich plasma Xiaomi generation can be used to study the low platelet count patients with low platelet count, for example, in thrombocytopenia patients, and also can be used to study the platelet dysfunction and measuring platelet-rich plasma Xiaomi generation can be also used to study the platelet drugs that affect Xiaomi generation and coagulation. Yeah.
Thank you. The second part of the question is also about reference values. In your paper on platelet-rich plasma, you showed the results of men and women together. Do you think reference values should be established separately for men and for women overall, or only for specific studies, such as those you done in PPP for estrogens contraceptives? So do you think we need separate reference values for men and women overall? Or
Yes,
does it depend on the type of study that you are designing?
Yeah, actually, there is a difference between the reference values between women and men, but I think it depends. It depends. In some situations, maybe we should consider the gender. But in some situations, we can just use all our reference values.
Okay, thank you. So I think these reference values are going to be very useful for studies in the future. And my last two questions are not specifically about your work, but rather imagining the future of thrombin generation and maybe thrombin dynamics. Do you think that the technique that is expected to be developed in the future is one based on point-of-care devices, POC devices? Do you have an experience of that question?
Yeah, actually, yeah, it can be used to point-of-care devices. There are already publications show that Xiaomi generation can be used in liver cirrhosis on point-of-care. And the whole blood Xiaomi generation is a good opportunity to use Xiaomi generation for point-of-care.
Thank you. And my last question is, do you think that the study of thrombin dynamics would be interesting in whole blood thrombin generation?
Yeah, actually, because the platelet plasma and platelet rich plasma also lack the blood cells in the plasma. So measuring the platelet, the whole blood thrombin dynamics is very good for clinical setting. So I think it is very interesting to measuring Xiaomi dynamics in whole blood.
Well, thank you. I have no further question and hand over the floor again.
Thank you.
Thank you, Professor Waal. And I turn to Dr. Baten. Dr. Baten is a postdoctoral researcher at the Department of Biochemistry of our own university here in Maastricht.
Thank you, Prorector. First of all, I would like to congratulate you with the nice work you have done and presented in your thesis. And also, I would like to extend my congratulations to the promotion team. My main interest is platelet function, so therefore I would like to discuss Chapter 5 with you. In Chapter 5, you assess thrombin generation and thrombin dynamics in platelet-rich plasma. You concluded that increasing the platelet count mainly affected the rate of prothrombin conversion and thrombin generation, but not so much the amount of thrombin that was generated. And further, you also showed that the total amount of thrombin that was converted increased with platelet count, but there was also a plateau reached at a platelet count of 150 times 10 to the 9 per liter. Also on page 113, you noted that the inter-individual variability in thrombin generation in undiluted PRP was not affected by the platelet count. And my question is, you have focused on platelet counts, but there will also be variations in platelet reactivity towards different agonists. So could you elaborate on how platelet reactivity towards thrombin, amongst others, fits within this process of thrombin generation and dynamics? And also, as platelets vary in size, do we need to consider platelet volume or platelet crit, parameter which both includes platelet count and platelet volume as a factor that can influence thrombin generation and thrombin dynamics?
Dear esteemed opponent, thank you for your kind words and thank you for your question. And if I understand correct, your question is, does the platelet volume or platelet drugs can affect Xiaomi dynamics and Xiaomi generation? Yes, we didn't study that in the manuscript, but I think the platelet drugs can affect the Xiaomi generation and the Xiaomi dynamics also, because the publication, there is a publication talking about that they found the Picomole, the Picomole can increase the ATP and increase the Xiaomi generation, decrease the ATP, so thereby decrease the Xiaomi generation. And also other platelet drugs, either platelet antagonists and platelet agonists can affect the Xiaomi generation. And for Xiaomi dynamics, because platelet can affect the pro-Xiaomi conversion and the pro-Xiaomi conversion. So I think platelet drugs can also affect the Xiaomi dynamic parameters. And for the volume of the platelet, for the volume of the platelet, we didn't study this actually, but there is published show that the Bernard-Soulier syndrome can affect can decrease the pro-Xiaomi consumption, and this syndrome is characterized by thrombocytopenia with gentle platelets. So I think maybe the platelet volume can also affect the Xiaomi generation and the Xiaomi dynamic parameters.
So if I understand correctly, it's a sum of all factors, reactivity, volume, and count.
Yes.
Yeah. In the discussion of chapter 5, you also mentioned contribution of platelets to thrombin generation other than providing a procoagulant surface. And here you mentioned factor 5. You state that factor 5 is within the alpha granules of platelets. They will be released upon activation. But when I read chapter 2 and I checked supplemental figure 3 on page 56, indeed, factor 5 shortens the time to peak and the lag time, but the effect is mainly noticeable at very low levels of factor 5, below 20% or maybe even below 10%. So my question here is, how can this 20% of factor 5 that is present within platelet granules contribute to thrombin generation?
Factor in the chapter 2, we measure the factor 5 levels within the increasing factor 5 level. You can say that is doesn't really make difference. Is one factor level is from 10% to 100%. So if factor 5 level is factor level is 20%. I think the Xiaomi generation and the Xiaomi dynamics would not get big differences. And 50%. Yeah, that is my.
So then the contribution of platelet factor 5 to thrombin generation and dynamics is fairly limited, if I understand you correctly.
Could you repeat again?
So the contribution of platelet factor 5 to thrombin generation and dynamics is then fairly limited, if I understand you correctly.
Yes.
Okay.
Yeah.
No, I'm afraid your time is up, Dr. Patton. I have to keep track of time, of course.
Thank you. Yeah. Thank you for your answers.
Thank you.
Thank you for your contribution. And I now give the floor to Dr. Huskens, and she's a research project leader at Synapse Research Institute here in Maastricht.
Thank you. Dear candidate, congratulations with your PhD thesis. And I also want to thank you for your nice and clear presentation. I also want to extend my congratulations to your supervisors. Thrombin generation can be a very useful tool to study coagulation. And in your thesis, you used a new technique first described by your co-promoter to get more insight in this thrombin generation. So I want to start with some questions on thrombin dynamics and more specific on the effects of fibrinogen and antithrombin. So you state that thrombin dynamics takes into account the measured thrombin generation curve, the fibrinogen level, and the thrombin inhibitors, antithrombin and alpha-2M, and that fibrinogen plays a modulating role in thrombin dynamics. So my first question is, how does fibrinogen influences thrombin generation and then also thrombin dynamics?
Fibrinogen, dear esteemed opponent, thank you for your kind words and thank you for your question. Regarding your question, fibrinogen actually play a modulating role on Xiaomi in activation. Fibrinogen can bind the Xiaomi and it can have affect its regular inactivation by anti-Xiaomi and alpha-2-myoglobin. So if fibrinogen level increases, you may get a higher Xiaomi generation and Xiaomi dynamic parameters.
Okay, and do you also know something about the fibrin polymerization? Does that play a role?
we didn't study that, but I saw there is a publication show that the fibrin variable can also play a role in the Xiaomi generation and the Xiaomi dynamics.
So does it then influence the prothrombin conversion, or is it something to do with thrombin inactivation? Do you remember that?
I think it can also factor the Xiaomi activation.
Okay. Maybe then I can go to my second question. So you describe that thrombin dynamics also takes into account the thrombin inhibitor antithrombin. Can you tell me which parameters of thrombin dynamics are sensitive for antithrombin? So you already mentioned, I think.
The Xiaomi decay constant is more sensitive to the anti-Xiaomi.
Okay. So then I want to go with you to figure 2K, page 37. There you look at the thrombin antithrombin complexes formed when you vary the concentration of antithrombin, and that the complexes stay constant. So the antithrombin concentration has no effect on the amount of thrombin antithrombin complexes formed. That's a bit strange in my opinion. What's the reason for that? Do you know that?
So could you repeat your question again?
So you would expect that if you have low antithrombin concentrations, that the complexes thrombin antithrombin formed would also be lower than you will have when you have high concentrations of antithrombin. But that's not what you observe. You observe a constant amount of thrombin antithrombin complexes.
If you understand correctly, your question is Xiaomi and anti-Xiaomi complex is not sensitive to the anti-Xiaomi level.
Yes.
Actually, the Xiaomi and anti-Xiaomi level is depends on the Xiaomi and the factor levels, as I mentioned in the presentation. Because I think the anti-Xiaomi mainly affect the rate of Xiaomi conversion instead of the amount of Xiaomi conversion, Xiaomi and anti-Xiaomi complexes is related to the portion. Xiaomi and anti-Xiaomi level.
Maybe I can help you a bit. In the discussion, you mentioned that it's maybe due to an excess amount of antithrombin relative to the amount of thrombin present.
Yeah.
But if that would be the case, then you should also can measure thrombin generation at low concentrations of antithrombin. So now you can only start measuring the thrombin generation curve when you have like 40% of antithrombin, because otherwise the amount of thrombin formed is too high.
Yeah.
So do you think this is then the explanation for?
Oh yeah, actually when anti-Xiaomi is lower than 40%, the pro-Xiaomi more than anti-Xiaomi in the plasma and causing ongoing Xiaomi generation. So the substrate consumption, we cannot get a accurate Xiaomi generation curve, so that we cannot see the the accurate Xiaomi generation when anti-Xiaomi below 40%. So that is might be a explanation why Xiaomi anti-Xiaomi complex is not sensitive to the anti-Xiaomi level.
Okay. I think my time is up.
Yeah, I think so. Yeah. Thank you, Dr. Huskens. Because I would like to give the opportunity also to Dr. Spronk. He's an associate professor of biochemistry at our university.
Thank you, Mr. Pro-Rector. Dear candidate, dear Qiuting, congratulations with this thesis, which I read with much pleasure. Also would like to extend the congratulations to the promotion team. To not lose any time on introductions, and I would like to extend on the question previously raised by Dana, and it is on figure 2 on the thrombin antithrombin levels, especially panel E and panel K. I did some calculations, which I normally like to do, based on the reference values for antithrombin, prothrombin. I first was intrigued by the observation, which was already raised in the first question, raised today, that the PC total, so the total conversion, is equal between the low tissue factor trigger and the high tissue factor trigger, or the mediate tissue factor trigger, the so-called 1 and 5 picomolar. If you look at figure C, all the prothrombin is converted into thrombin. There is almost no prothrombin left. That means that if there is 100,000 nanomolars or 1 micromolar of thrombin formed, that in figure E, there is also 1 micromolar of thrombin antithrombin formed. The reference value for antithrombin is 2.6 micromolar, meaning that almost 40% of the antithrombin is bound to thrombin. Then looking at panel K, at 40% antithrombin levels, you still have the equal levels of thrombin antithrombin levels. So all antithrombin there is bound to the enzyme. And I would like to hear your opinion on that. How is that possible that if you have 40% of antithrombin, everything is bound to thrombin, and if you have 100%, still 40% is bound to thrombin? For me, that doesn't make any sense at all. To raise the stakes a little bit as last question, but would like to hear your opinion on that.
Dear esteemed opponent, thank you for your kind words and thank you for your question. If I understand correct, so could you repeat your question again?
I can rephrase it a little bit.
Yeah.
The first part is, how is it possible that 1 picomolar of tissue factor and 5 picomolar of tissue factor give completely the same equal prothrombin to thrombin conversion? That's panel C. So it doesn't matter how you activate, with what you activate, it's always the same. How is that possible? Yeah. And then doing some basic calculations, it's always 40% antithrombin that is used to completely inhibit the thrombin formed, meaning that in panel K, at 40% antithrombin levels, no antithrombin can be used to inhibit factor 10A, to inhibit factor 7A, to inhibit factor 9A. It's only inhibiting the thrombin. And that's something I don't understand. All the other enzymes must also be inhibited by antithrombin, especially factor 10A.
So your first question is why the 1 picomolar tissue factor and 5 picomolar tissue factor have exactly the same results on Xiaomi dynamics. Yes, actually we have the similar results on 1 picomolar and 5 picomolar tissue factor.
Let me then rephrase it for you in another way. to get the discussion going. You nicely demonstrated that if you lower factor 8 or lower factor 5, that you then get a difference in the PC total. So if you drop in the coagulation factor below 20%, you get a difference in the PC total, but not in the activation. For me, that was a big surprise to see that in this way. I never looked at it in this way. And that led me thinking how that would be in the human body with thrombosis. So it doesn't matter how much you activate, the amount of thrombin generated is always the same, unless you have a coagulation factor deficiency. And that surprised me a little bit, and I don't have any explanation on that, to be honest, but I would like to hear your opinion on that, on this finding.
Actually, with low coagulation factors, we have a low Protrom conversion and Protrom conversion. For example, in hemophilia A patients that I showed in the on the page 5, we really have the low Protrom conversion and the Xiaomi activation. And for the different level of tissue factor, with different tissue factor levels, because for one Picomole tissue factor, it activates the intrinsic coagulation pathways.
And the beetle calls. When the beetle calls, I'm afraid the opposition is over. So I thank you for your contribution now, but Mr. Qiuting Yan, the time appointed for the defense of your thesis now has ended. The degree committee will withdraw to discuss the quality of your thesis and of the defense you just gave, and I would ask you and your company to await the results of our deliberations and our return to this room. And thereby we are transferred to our private room. here online has assessed the quality of your thesis and of your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor ten Kate, your supervisor, is authorized to confer upon you this academic distinction in accordance with Dutch university custom, so I invite your supervisor to take the floor.
Thank you very much, Mr. Pro-Rector. Dear candidate, we have to start with a promise you have to make. And that is the following: do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible? I can see your lips moving, and I assume you say yes, but maybe you're still muted.
Yes, I promise.
Thank you very much. By the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer upon you, Qiuting Yan, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor, affixed with the official seal of the university, as now shown by the beadle. So, congratulations, Dr. Yan. I'm happy to be the first one to congratulate you with this achievement on a long distance. You did very well, and enjoy this great honor and appreciate it. And I hope you have a small celebration later with your family and friends. But for the formal laudatio, I hand over now the microphone to Dr. De Laat.
Thank you, Professor ten Kate. Dear Dr. Yan, dear Qiuting, congratulations with successfully defending your thesis and therefore obtaining your doctor's degree. On behalf of the promotion team and of Professor ten Kate and Dr. De Laat, I would also like to congratulate your friends and family with this occasion. Qiuting, what a trip you made, both physically from China to Maastricht, but also personally from a very young PhD student to a grown-up scientist. I know exactly how you started here in Maastricht. We did an interview via Zoom or Teams, and you were very timid. Together with the promotion team, we discussed, is she capable of conquering the scientific world, or maybe even surviving the scientific world? But you did really well. You proved it otherwise. Indeed, you were very timid, but I think modest is a better word. And you proved to be smart, capable of learning at a very fast pace. This really helped you during your PhD period. For example, statistics. You've been working with large databases, over 20,000 patients, over 50 parameters, and also some smaller databases with more complex mathematics. But you arranged your own statistic courses, and in the end, you came up with tables and manuscripts, and we checked, and everything was correct. So that was really surprising for us. And you also enjoyed working in the lab a lot, often asking for more experiments to do. For example, when we ask you, can you measure this parameter in 23,000 samples? You just said, yes, of course I can do that. So that was not a problem at all. And every day you came into the lab, always fashionably dressed, and continued your experiments effortless until you collected all your data. Working with you was a pleasure. We always had positive discussions about data, results, and the interpretation. Sometimes I felt you didn't understand me due to probably the language barrier sometimes, but days later you came up with the results that we discussed, always with a genuine smile. not only the staff of Synapse like you, but yeah, we've had a lot of PhD students from China, and you all were a really nice group to have within our institute. It was a pleasure to host you and to teach you all the things about science that we could. You also worked very hard and long hours. I remember sometimes I went to the laboratory, I turned on all the lights, and from some corner I heard, Bas, can you turn on the lights again? I'm still working. So that really showed that you were willing to put all your best to obtain your PhD degree. The last few months, you were sometimes a bit worried that you would not succeed finishing your thesis, but due to your persistence and hard work, I think it's a very beautiful thesis that you have presented today. Well, Qiuting, finally you can relax. You were really nervous for today, but you can take a deep breath. Again, congratulations with obtaining your doctor's title. Well done.
Thank you. Thank you very much.
Yes. Dear Dr. Yan, you saw me applaud. That was a bit for your promoter, but it was also for yourselves. On behalf of the Board of Deans and the whole academic community here in Maastricht, I would like to congratulate you with the honor that you've acquired. And that congratulation, of course, also goes to your supervisors, so Professor ten Kate, Dr. De Laat, Dr. De Laat Kemers. I would like to thank the opponents, especially our guests, as has been done earlier by Professor ten Kate, but Professor van Gorp and Professor Waal, it was really an honor to have you here and to see you contribute. I'd also like to thank the Chinese Scholarship Council, who made this possible, of course. We have a good cooperation, I think, with the CSC. Dr. Qiuting Yan, I wish you all success in your future career and hope that you will indeed have the time and the opportunity to celebrate with everybody around you this doctorate that you achieved today. Congratulations again, and thereby I would like to close this session.
Okay.